Mr. Enzyme, I think you're a pretty cool molecule. Can I get your number? Your enzyme commission number, that is. That way, I would know more about you, and I could look you up in this ExplorEnz database to find out more about your hobbies, the reactions that you catalyze. So, all puns aside, the enzyme commission system, like nomenclature and these like EC numbers and these systematic names, this is all like super duper important because it's really important that we be able to distinguish between different enzymes and know what the heck people are talking about. People need to be able to know, is the Joe that he's talking about the same as the Joe that she's talking about? Is that old yellow enzyme the same as the new yellow enzyme? And so I was joking about those Joes, but I am totally serious about these yellow enzyme business. So old yellow enzyme is AKA NADPH dehydrogenase. New yellow enzyme is AKA D-amino acid oxidase. And so these are totally names that you would read about in papers, and people need to be able to know, okay, well, what enzymes are actually like the same, what are these different. People were discovering the same enzyme, naming it different things, calling the same enzyme different, or calling different enzymes the same thing. All sorts of confusion, all sorts of chaos. Speaking of chaos, there are thousands and thousands of enzymes. So we need someone to bring order into this chaos. We need an international committee. We need the IUBNB nomenclature committee responsible for the enzyme commission numbering system and their database ExplorEnz. So let's talk about how this database works and how these enzymes are given these unique IDs, these EC numbers. Just a super duper quick note on enzymes, much more in other posts, and then we'll dive into how they're given these names and numbers and how we can find more information about them. So enzymes are biomolecular reaction speeder uppers, and so they're often proteins, sometimes protein RNA complexes, sometimes RNA alone. Often they're working with the help of like cofactors, so little like helper molecules like vitamins or metals or that sort of thing. And there are a ton of enzymes because there are a ton of reactions that need catalyzing. Like if you think about all the stuff that has to happen inside of all your cells, and then you remember like, whoa, I'm just a person. There's like all of these other organisms around me, and some of these things, like these little microbes, are doing things like crazy little things, like who would have known? So basically there are tons of reactions and tons of enzymes, and we need a way to tell them apart. So before we get started, I want to thank Drs. Keith Tipton and Ron Caspi of the Enzyme Nomenclature Committee, who helped me with guidance and fact checking and all sorts of stuff, as well as Dr. Alexandra Newton, president of the IUBNB, who put me in touch with them. And I have this whole long post on my blog, totally new content, and figures and stuff, a lot more about the different classes and the classification schemes. So I encourage you to check this out, but I also wanted to give a quick video to help kind of walk you through things, as well as show you how you can use this ExplorEnz database to find out more information about various enzymes. And it also gives you a good sense about what the different classes and subclasses and all of this means. So these are the EC numbers that we are going to be talking about, which is just part of the information that we'll be talking about, actually, because these are key to kind of getting to the ExplorEnz entry, where you can find a lot more information, such as accepted name, systematic name, what reaction they catalyze, and various things like that. And we'll get much more into this and more into details. But first, I want to just start off by saying that naming enzymes is hard, or numbering enzymes and classifying them is a really hard job. And this is just, so this database has grown greatly, and it's still it's a living system. And so they accept nominations for new enzymes to be added to the database, and people can submit nominations, and then they go through a strict process, and there's like all of this review stuff. And it's not just like a People's Choice Award. They make sure that all of the information, that these enzymes are truly unique, and the reactions that they're catalyzing are like real, and there's like strong evidence for them. So with these EC numbers, this is kind of going to be
your ticket into finding out more information, and we'll see that there's a lot more information that we can find out in these ExplorEnz entries. But these numbers are these are the like identifiers. So in these numbers, there are 4 parts. And so the first number is going to tell you the main class. And so the first number is one of 7 numbers. So there are 7 classes of enzymes. So there are 7 classes of enzymes. If you look at earlier stuff, you'll see like 6 enzyme classes. The 7th class, the translocases, was added in 2018 and stunned the world. So a lot of these enzymes were already in the database, but they were categorized in different places. And so then they got their own category, these translocases. These are things that are moving things from one side of something to another. Typically, these are some sort of transporter across a membrane. They're often associated with some sort of enzymatic activity reaction. So maybe you have some sort of hydrolysis or carboxylation or something that's coupled with the movement of things across the membrane. And so this isn't just like a channel that's open, it's actually actively doing stuff. The other classes, we have the first class is oxidoreductases. These are catalyzing a redox reaction, so oxidation and reduction. Things where you're moving electron. There's the movement of electrons or electron density, much more in other posts. If you see a 2 in the first place, that's going to tell you this enzyme is a transferase that's transferring a functional group, such as an amino or a methyl group. 3, we have hydrolases. These are using water to break bonds, so they're catalyzing hydrolysis. 4, we have lyases. These ones are kind of confusing. They add or remove things to make or break bonds. These are often double bonds, but not always. The key thing is that they're doing this in a way that isn't hydrolysis, and you're not using hydrolysis, and it's not using redox. So these are classified as lyases, and so they often involve like double bonds or ring structures. 5, if you see a 5 in that first place, that's an isomerase. So it doesn't look like anything's happened here, but what's happening is that these are actually rearranging things in the molecule. So maybe shifting things over a group. So maybe this group was sticking off of this carbon and now it's sticking off of this carbon, or changing the stereochemistry. So maybe you have an L amino acid and they change it to a D amino acid. So kind of like changing the handedness of things. 6, you have ligases. So these are joining together molecules, often with the help of ATP or another nucleotide triphosphate, that's providing the energy for that. And then 7, that was that new one that we were talking about. So the first number is the only number that you can actually know, like you can look at it and say, without looking anything up, if you have these 7 classes memorized, you can say, okay, I know information about this enzyme right away. If it starts with the 1, boom, it's an oxidoreductase. Starts with the 4, now you're looking at a lyase. And so this can be helpful. And not that the other numbers aren't helpful, but this one, you don't have to look anything up. You get information right away from here. The subclass and the subclass, these are going to further classify things within this main class. And what they represent is going to depend on what this first number was. So if you had like a 1 here after a 1, it would mean something different than a 1 here after a 2, because these are subclassifications within this main class, but the types of classifications that you'll have are going to depend on the main class. And this will hopefully make more sense when we look at an example. The subclass and the subclass are telling you typically more information about the reaction. So maybe they're telling you what the substrates are. Maybe they're telling you what types of bonds are broken, what types of bonds are made, what types of various things like that. The serial number, this is not providing any like information. It's more just like an ID, so that every enzyme has its own unique EC number. So now we need to get into a little complication, this complicating factor of what an EC number actually represents. And what do we mean? Like, who gets a unique number? So when we're talking about these EC numbers, it can get kind of confusing because we need to be able to kind of like change our mindset as what these numbers are representing. So we could have the same kind of like overall reaction. So you can have A plus B goes to C, happen in different ways. So you can have a DNA ligase, say, so it's going to stitch together DNA. So you take 2 strands of DNA and you can stitch them together. But some DNA ligases, like the ones that we normally think of, are using ATP to do this. Some are using NAD+. There are other instances where some things use ATP, some use GTP, some might catalyze a reaction with oxidation or reduction. Others would do it with hydrolysis. But in all of these cases, you have it where you have the same end product and you have the same like overall reaction, but it's happening in different ways. And because you have different like real reactions actually happening, then you are going at the atomic level, what's actually going on is going to be a different reaction. And so these are going to have different EC numbers. We'll get into an example where we're going to see this in our ligase case. Another thing is that one enzyme or complex with multiple active sites doing different things can have multiple numbers. And so you can imagine you have some big complex. Often these will say like complex or system, or they have some in parentheses in the name. If they're doing different things, these will be given different EC numbers. This can be helpful too in the case that sometimes in different organisms, there'll be a complex where like different chains are actually doing different things. So one reaction is going to be carried out by this chain and another one is going to be carried out by this chain. And then overall, you have these 2 reactions happening, but then in a different organism, maybe those genes fuse together. So instead of a single chain, instead of 2 chains, so instead of having something like a dimer, now you just have a single chain, you have a single protein, but it's carrying out these 2 different things. And so you would still have these 2 different EC numbers. And this could be helpful if you have in those cases where you can distinguish between if it's like the same activity that's happening, it just happens to be on 1 chain versus 2 chains, versus if it happens to be like 2 things that happen on the same chain versus the same thing that's happening in a totally different subunit in a different complex. This way those things could still have the same EC number, but then this one complex would have 2 different numbers. Speaking of things having the same number, so isoenzymes, so things such as homologous proteins. So if you have a protein, you have an enzyme that's catalyzing the same reaction, but it has a different like gene associated with it. And so in our body, say you might have, we're just going to call this like cool enzyme. So we'd have like cool enzyme alpha, cool enzyme beta, cool enzyme gamma, or maybe you have cool enzyme 1, cool enzyme 2, cool enzyme 3. And often these come from a gene duplication event where the gene was duplicated and then evolution kind of tinkered with it. And so they get slightly different protein structures. And so they might have, like one might have a longer, like an insertion or some sort of a longer tail or something that might be regulated differently. But at the end of the day, they can often provide like backup for the other ones. If you knock one out, you don't even see an effect because these are catalyzing the same reaction. That doesn't mean it's not important that you have different versions of them and that you're able to regulate them differently and all of this various stuff. But in terms of their enzyme commission number, they're going to have the same EC number. So in addition, that same enzyme in a different species, so the orthologous version, so like the mouse version of cool enzyme 1, it's going to have the same EC number as the human one. There are also cases where you could have enzymes that are doing the same reaction, but with slightly different like specificities in. And when we talk about these like specificity differences, we're talking about like big specificity differences. We're not talking about, so we'll see with restriction enzymes how they have the same EC number because they're all cutting in the middle of, they're cutting in a sequence specific, they recognize the sequence and they cut in the middle of DNA. And it doesn't matter that the sequences that they recognize are slightly different. That's not big enough of a change in order to classify them as a different type of enzyme. And so you'll see that enzymes can look totally different. And the thing is, like with this EC number, I see when I think about enzymes, I'm thinking about like individual molecules. So I'm typically talking about enzymes or individual like complexes. Or basically, you have this it's a physical enzyme, but these numbers are talking about the reactions that are actually going on. And so you have to think in that mindset of the reaction that's going on when you're thinking about these EC numbers. And when you actually go into the ExplorEnz database, you can find more information about this reaction that is actually going on. And so I talked about restriction enzymes yesterday. These are sequence specific DNA cutters, super duper useful in the lab. And basically all of them have the same, all of the ones that we conventionally use, typically think about these like type 2 restriction enzymes. The classical ones we talk about, there are other types of restriction enzymes that have different numbers, but these type 2 ones that we talk about, like BAMH1 and all of that, these are going to have this same restriction number, EC 3.1.21.4. And so this number, you might think, okay, well, what the heck is that telling me if all these different enzymes have the same number? But this number really does provide you information. It's just the serial number might not be the same as what you would think of as the serial number for a given enzyme. Like, it's not taking you to the final. There is no molecule exactly like this one. It's saying that there is no other, there's no other class of molecules kind of that carries out this reaction in this way. And so that is the endpoint that we're taken to with this number. And in this number, we get information that's taking us to that point. So all of these numbers are giving us information. And so the three, this is telling us that it's a hydrolase. So that refers to the class, remember? And so this is the number that we can look at and know things without having to look up the tree and see what the next numbers correspond to. This one is going to tell us, I'll show you how we can find out this stuff. This one tells us that it's cut ester bonds. And so this is an ester. And so this one is basically, this is the main class, this is the subclass. And so the subclass in this case is telling us about the type of bonds it cuts. Then the 21 is telling us more about that. So it's saying it's an endodeoxyribonuclease. So this is telling us about the type of bond, and this is telling us about like where that bond is located. And so this bond is located in the middle of a DNA strand. And it is going to be cutting this strand, and it's using water to do so. And so that's why it's a hydrolase. And then the 4 is the serial number that's telling us it's a type 2 site-specific deoxyribonuclease. How I like to go and view this thing is by going in the ExplorEnz database. And it's kind of like going on a choose your own adventure enzyme style. So I'll take you on a live example in a minute, but what you could do is basically, if you go to this enzymes by class page, you can expand these different trees. And I like doing it this way because it allows you to see what is nearby. What other things are in the class, what is in the subclass, and then you can click on the links to get information about what qualifies you to be in that subclass or be in that class. And so you can see that the other things in this class are going to be like other types of restriction enzymes and that sort of thing. So there are similar things around it, but this is going to be the unique entry. So I'll take you more on a live tour of ExplorEnz in a minute, but first let's talk about the kind of information that we're going to find when we actually go into its entry. So this is from this really great article that I recommend and that I will link to, and it gives you a lot of information about the nomenclature committee and the whole process and this ExplorEnz database and what you'll find. So we've been talking about this EC number. And so this EC number is that 4-digit number or 4-component number. And it also gives you names. And so the accepted name, This is usually the most commonly used name for the enzyme, as long as it's neither ambiguous nor misleading. So this is basically what you would normally probably be talking about in terms of when you were talking about this enzyme. So the EC number are super duper important. And when you have a paper or that sort of thing, it gets connected to the EC number. And so that way, if people are talking about this enzyme, you'll be able to know this is the enzyme that they're talking about. But when we're referring, and you can cross-reference between them and that sort of thing, but in general talking, we're typically not going around talking about 3.1.21.4. We're going around talking about that type 2 restriction enzyme, restriction endonuclease that we used or whatever. And so that would be the accepted name, would be how people generally refer to it. There's also often a lot of other names that people have given to this enzyme. And so in the case of our old yellow enzyme and the new yellow enzyme and stuff, those would show up in the other names, not in their accepted name. Even the accepted name, though, this might be kind of confusing or might not be as informative as you would hope, because you can't give all the information that you need in a name that you would use in normal conversation. And so then you have a systematic name, which is going to describe the reaction catalyzed in unambiguous terms. So this can get kind of long and unwieldy, but they will give you the most information and be the least ambiguous. A lot of times with these other names, you'll see a lot that have like in parentheses ambiguous or like wrong various things, but they want you to be able to know that people also have sometimes called it this. With the accepted names, it can get confusing because sometimes these accepted names are not necessarily how the reaction normally goes. So because enzymes can catalyze reactions in both directions, and in our body, they might catalyze they might really go both directions. And so we have some of the same enzymes used both in anabolism, so molecule making, and catabolism, molecule breaking. And so we have some steps that are reversible when we're talking about enzymatic like metabolic pathways, such as glycolysis, to the breakdown of sugar. You can see that a lot of these steps have these arrows going in both directions, showing that these steps are reversible and they are easily reversible. And depending on like how this is regulated and the concentration of the various things helping drive the reaction in one way or another. Other times, however, the arrows, so in the case of one of these like easily reversible reactions, then you have to think about, okay, well, if it's doing this reaction in both directions, how do we decide what to call it? And so often these names are what people the accepted names would be what people originally, what they discovered that it did like first, not necessarily what it always does. And then in terms of the reaction catalyzed and that sort of thing, they have it so that the enzymes in the system, in the entries, they have it so that everything in the class the reactions are going to be written in the same direction. Not necessarily, that doesn't mean that that's the direction that it normally like physiologically goes in. It's just consistency with the class. And so it might be opposite of what actually normally happens if just in how it's written, but know that enzymes catalyze reactions in both directions. So for example, if we have phosphoglucose isomerase, well, it could also, you could think of, well, isn't it a phosphofructose isomerase? And so you can see where the naming can come in. Sometimes names are just super duper confusing, and for reasons like that just have to do with how they were originally named. So an example of this is like pyruvate kinase. And so this carries out this key step in glycolysis. This step, you have a forward arrow, so this is basically irreversible. And it's converting phosphoenol pyruvate into pyruvate. And this step, the enzyme that catalyzes this is called pyruvate kinase, although this is an irreversible reaction, so you're not actually having it add a phosphate group onto pyruvate. It was just a historical name. And so that's one of these names that often confused me in undergrad. And so no, that's a good reason for why we have these systematic names as well. And we are able to look up the reaction diagram and see and often, so this is a good reason why we have these systematic names that are unambiguous. And then you also have in the comments, they often tell you about which way is normally favored in cells, or how it sometimes they're like organism specific. If only like weird microbes have been found to have it, they might tell you about that. Sometimes in the name, you will see information in parentheses. So often they'll have information about like cofactor requirements. So maybe it needs a vitamin to work, or that sort of thing. And so you often see these in parentheses, as well as maybe it needs ATP or NADH or various things. And so you'll see those in parentheses. And sometimes you'll see that different versions, if you have different like enzymes that catalyze the same reaction in different ways type of thing. So you can have a situation where you have like a DNA ligase, if it uses ATP, it'll have in parentheses ATP, and then a different EC number will be given to a DNA ligase that uses NAD+. And so here you're seeing an example where you have information given in these parentheses, providing even more information than the number alone, so you can sub-classify between them. They tell you about the reaction that's catalyzed, diagrams, when sometimes they have links to diagrams, very cool stuff, links to other databases and references, as well as the history. So if it's been moved from a different number or a different category, that sort of thing. When this happens, so sometimes they create new categories, like we see with the translocases, they had a whole main class, new main class. Other times it's more just like subclasses and that sort of thing. If an enzyme is changed, if its number is changed, the old number won't be assigned to anything. And if you go to like try to look up the old number, it'll take you to the new number, but they're not going to give a different enzyme that number. And so it'll be like retired or whatever, so that you don't get confused there. Speaking of DNA ligase, so DNA ligase is going to join together a broken DNA strand, or where you have a free 3' OH and a free 5' monophosphate. And in the case of the DNA ligase that we normally use and that we're going to be talking about in this entry, it's going to use ATP. And so we call it a ligase because it's using this triphosphate, this nucleotide triphosphate, or a similar molecule. In this case, it is using ATP in order to get the energy to join these together. And how it does this is cool, and I have posts on that and that sort of thing. But that's not what we're going to talk about today. Today, we're going to talk about how we can look it up in this ExplorEnz database. Typically, if you Google an enzyme, there are various ways that it'll like show its EC number. Sometimes these EC numbers will be linked to different databases. And so you might get linked to like a Brenda database or something like that. Those databases are not the official database. The official database is the IUB&B nomenclature one. They're like the people in charge of all of this, and they have that strict vetting process for the different enzymes and make sure that everything in there is like super duper secure and all that, up to date, and like verified, because there, you can imagine that there's a lot of different activity. It's hard to kind of tease out what enzyme's doing what, and are these enzymes really different. If you're using a different database and it has like an extra number or it has a letter at the end, those aren't the official numbers. Those are the database trying to add some additional information, but that information has not been like verified and validated by this database. I'm going so in depth into this because I got confused about that too. And so Drs. Cassidy and Tipton were really helpful with that. And so I just wanted to bring that up. because you often get taken there, but you can also find all of this. The ExplorEnz database is really the place to go. And typically, like if you go to Brenda and stuff, it'll have a link to the ExplorEnz entry. But as I'll show, I like to go in this other way to look it up by this enzymes by class. Okay, so this is the ExplorEnz database. So this is the official IUBNB nomenclature committee database. You can find a lot more information about various things, see what's new in the world of enzymes. I like going to this enzymes by class, and this is where we get that choose your own adventure. So if you go here, so these are the 7 main classes, and if you want information about them, you can click on them. So we're going to be looking at a ligase, and it's going to say ligases are enzymes that catalyze the joining of 2 molecules or 2 parts of a molecule with concomitant hydrolysis of the diphosphate bond and ATP or similar to triphosphate. Ligase is often used for the common name, but in a few cases, synthase or carboxylase is used. Synthetase may be used in the place of synthase for enzymes in this class. Cool. Okay, so now we know what that is. We're looking for 6.5.1.1. And so if we go and we press on this plus, we can expand this ligase category. So remember the 6, this is our main class, and that's going to be telling us it's a ligase. If we go to 6.5, now it says, okay, 6.5 corresponds to enzymes that form phosphoric ester bonds. And so if I were to click on this, I would get information. Okay, so this is what is involved in here. This subclass contains enzymes that restore broken phosphodiester bonds in nucleic acids, often called repair enzymes. And then it's telling us that there's a single subclass. And so if I then expand this class, so that was the class, and then this one's a bit of a anticlimactic, is that there's only a single subclass in this case. But you can see that the 6 was telling us it was a ligase. So the next number, the subclass is telling us what type of bonds it's acting on. And then if we expand this further, now we have the serial numbers. And so this is our last number. And here, if you click on these serial numbers, then you're going to be taken to the entry for that. And so you can see that we have the accepted name, where it's telling us that it's a DNA ligase that uses ATP. We have the reaction that it catalyzes, and we have other names. And so you can see how many of these names, you have these ambiguous names like DNA joining enzyme. I like deoxyribonucleic acid joinase. There's a lot of names that sound like they would be very good wizarding spells, but not so great with talking about enzymes. There's also some cases where you have really cool names that sound like they'd be really cool superhero names. But anyway, so this is its systematic name. And so you can see that it's long and unwieldy, but totally unambiguous. But you wouldn't want to be talking about this in daily use. So if you think about how long and dry scientific papers can sometimes seem, imagine if every time you wanted to talk about DNA ligase, you had to write out this whole thing. But you can imagine that there are cases when it's really important to know all of this information and to be able to specifically differentiate between what enzymes people are talking about. You can see that in the comments, it's going to tell you more information about it. So it's telling you more about its reaction mechanism. It's just cool. You'll see different information in the comments for different enzymes. You can get links to other databases. So PDB, that's where you can look at protein structures. There's some stuff with metabolic cycles. Brenda, so this is an enzyme database. Not this official enzyme database, but it has information, different types of information and stuff. And then you'll also see references that you can go find more information on. So I like to find the entries by the enzyme by class so that I could go through that function where I could expand all these different things and find more information about why it's given these various numbers. So to drive home this point that only the first number is really going to give you information without relying on any other outside information. So we saw with our ligase that we had 6.5. So our 5 was telling us that it was forming these phosphoric ester bonds, and then the 1 was just because it was the only one in that subclass. But if it was something within a subclass where there were further differentiations, then it would tell you more in that class. But these numbers are going to be specific for a ligase. And so we had a 5 here telling us it was forming phosphoric ester bonds. But if we go and we look at, say, a transferase, now a 5 is telling us it's transferring alkyl or aryl groups other than a methyl group. So now it's telling us about the type of group transferred. And so you can see that then, and within that, you get further well, that one you again only have a single class, but if you were to go to glycosyl transferases, now you get more information about hexosyl. So the types of basically these types of sugar, these sugar chains that are being these sugars that are being transferred. And once again, in each of these cases where you see a link, where you see something blue, you can click on it and look things up. And you can see how many different enzymes there are. Woo, this is just like in this one subclass. And so lots and lots of enzymes. And so this is really cool. But these numbers are telling you different things depending on the context of the numbers that came before them. So I highly encourage you to just like play around with the site, even if you don't know a specific number that you want to go to, you can kind of go on an adventure and see the different types of enzymes that there are and what gets classified where. Click on the links to find more information, various things. You can see what are the new and amended enzymes. And this is showing you this NC IUB&B committee, so the nomenclature committee of the IUB&B. And if you have comments and suggestions on these draft entries, so these are proposed changes to the list. So I told you they had to go through this big process. And so these are all proposed changes. So when people propose changes, then they go through the committee, and then they go through a public review. And so very thorough checking. And then they'll eventually get added if that all pans out. And then they have forms where you can have a new enzyme form. So if you want to propose a new enzyme, you have to tell them why and all of this various stuff. And then it will be sent to the committee where they could review it and see if they want to give it to the public review and all of that. You can also download all of this various data. So really cool stuff. So I really hope that you will go and explore it. And thanks again to Keith Tipton and Ron Caspi for helping me understand all of this and giving me a behind the scenes look at kind of answering all of my questions and telling me cool history stories. So thank you to them, and thank you to the nomenclature committee, because they're a volunteer committee. They are doing the really great work without a lot of people don't really usually get that excited about nomenclature and that sort of thing, but we rely on it and it's super duper important. And so this work is super duper important. And so thank you to them all. And thank you for Enzymes for being so cool that we have to have all of this stuff to keep track of them.